This is our continuing discussion of Simondon's Individuation in light of notions of form and information. Chapter 1 of part 2. I can't remember which section we're on, but we're on subsection 4 of that section. We're going to start from page 199 today. What we've been seeing so far in this part is the way that Simondon connects the notions of the individual and reproduction in vital individuation. And this is based on the analysis that he's given of these types of organisms that have both a colony form and an individual form. There are certain corals and polyps and things like that. I think they're all of the organisms that have these two forms are aquatic organisms, I think. So you have these polyps or whatever the organism is that live in this colony form, in which the multiple organisms share a circulatory system and other functions. And so in some respects, they make up one individual. The whole colony is a sort of quasi-individual. But then, and in that form, they reproduce just by budding. They split off to reproduce. Then there's also this individual form. So depending on which type of organism, there are certain conditions under which one of the organisms will separate off from the colony and have a free-floating or independent existence for a certain period of time until it sort of refounds a new colony in a new location and then reproduces through budding again. So Simondon takes these two forms, the colony form and the individual form, as corresponding to two different functions in the sphere of vital individuation. And so even though there's only certain types of organisms that have these two forms, we can sort of look at analogous functions in other organisms and compare these colonial type functions, if we can use that term. So the functions that are characteristic of sort of everyday life and growth and metabolism and so on. And then on the other hand, you have these functions that are characteristic of the individuals as individuals. So reproduction, and then as we've also seen, death is characteristic of individuals. So the individual organism is born, it reproduces, and then it dies after a more or less determinate lifespan. And that's part of what's characteristic of an individual in the vital sphere is that capacity for death. And in subsection 3, the last part that we read, it was looking at specifically the different modes of reproduction that you have in different organisms, and contrasting schizogony and gamogony. So schizogony being reproduction through fission or splitting of the organism, which many unicellular organisms reproduce in this way. They one cell will split into two, and then those two cells go on to have independent existence. And then gamogony being a form of reproduction in which you have two cells that have to join together first before they again split to form new individuals. So this is sort of a one step along the path towards sexual reproduction, but in many organisms there's no differentiation of male and female, and so you have two individuals that are basically the same in structure that fuse, and then they split twice and form four new organisms. And so Simondon, looking at the various forms that these two modes of reproduction take on in different species, he looks at the ways that these two forms of reproduction can be joined together in different organisms. So there are some organisms that have both forms of reproduction in alternation, so that one stage of the organism, for example, lives in human blood as a sort of amoeba-like creature, and then if it gets taken up by a mosquito, then it reproduces asexually and produces these spores, which the mosquito then deposits in a new human host. And then it goes back to the amoeba-like form and reproduces sexually, or in the gamogenous form. You can have both forms of reproduction can occur in the same species in alternation, and sometimes there are even sort of complicated combinations of the two forms where there's like a fusion and a splitting that happen together. But the sort of takeaway that Simondon derives from these various forms of reproduction is that we can't make any sort of substantial distinction between the two functions that he argues are characteristic of living organisms or of vital individuation. So the colony functions and the individual functions can't be sort of split off into two separate substances in the way that Weissman argues that you have a soma and a germen, which are two distinct substances which have no real interaction with each other. So the fact that an individual cell, or even in certain multicellular organisms, the whole organism can reproduce through splitting, shows that there's no sort of substantial distinction between the reproductive matter or the reproductive substance in the organism and the substance of the soma, or the sort of everyday life aspect of the organism. So the distinction between these two functions can't be reduced to a distinction between two distinct substances, is sort of the message from subsection 3. And that's about where we left off. So we'll pick up from there in subsection 4. Okay, so I'll start reading and then we'll go around as usual. Yeah, subsection 4, Undifferentiation and D Differentiation as conditions of reproductive individuality. Due to a sort of law of opposition that appears in every problem concerning the individuated being, what the individual gains in density and in substantiality when reproduction is defined as a regeneration and not as a transmission of the germen from soma to soma, it loses in independence relative to other individuals. Those species in which the individual substantiality is most solid and obvious, going all the way to the capacity of never dying, insofar as each individual can divide without remainders, are also those in which the individual's boundaries are the most difficult to trace, because all the modes of association exist in it, and because reproduction often gives rise to intermediary forms between an organism and a society, forms which are hard to name insofar as they are basically mixtures. This disappearance of the independence of the individual can either occur provisionally in budding, or definitively, thereby leading to a colony. Even in the colony, various degrees of independence are possible. Budding produces independent individuals, but it only produces them slowly, and the various fragments first proliferate before separating from one another, as if regeneration were consequently prior to schizogony instead of following it. This preliminary regeneration via proliferation gives birth to a mass of indefinite contours, which protrude weakly at first, then increasingly, and which is then called a bud. Amplification is contemporaneous with the beginning of the process. The region in which the bud occurs is more or less narrowly localized in general, which, according to Rabot, does not imply a special type of properties, opposing it to all the other regions, and which would designate it as the support of a possible germen. Localization certainly arises from some secondary arrangement that reverberates throughout the local metabolism. Localization is a secondary incident. It is necessary to merely remark that the parts of the body most capable of detaching and proliferating, like the hydra's tentacles, which play the role of the germ, are not the bud's birthplace. On the contrary, the outer lining of the body is what easily produces buds, which eventually separate from their point of origin. According to Rabot, only local, purely contingent conditions would be able to restrict an absolutely general possibility in its essence to certain elements of the body. This possibility of proliferation would not be the privilege of certain elements of the body to the exclusion of certain other elements. What the two modes of reproduction have in common, budding and schizogony, is the existence of undifferentiated and de-differentiated elements that play the role of reproductive elements while remaining unspecified elements of the body. Before the proliferation of the schizogonic germ, like at the moment of a bud's formation, the elements that prepare the formation of this germ or of this bud conserve or recuperate embryonic properties, i.e. remain undifferentiated or dedifferentiated. The localization of budding and its essential characteristics manifest in coelenterates of the hydrozoan group. In the hydra, the bud is at a diverticulum of the outer lining that spreads out, expands, and then pierces its vacant extremity, from which tentacles appear. The bud seems to originate from undifferentiated cells that actively multiply and insinuate themselves between elements of the endoderm and between those of the ectoderm for which they are substituted. These cells would therefore not be dedifferentiated but non-differentiated. They play the role of veritable generative cells. What would give birth to this localized budding would be these cells' distribution under the integumentary epithelium due to unknown influences. The substantiality of the whole individual would be absolutely certain if it could be confirmed that dedifferentiation is the sole procedure of budding. It is less clear in the case in which, like in the hydra, there is an undifferentiation in question. But Rabot points out that these undifferentiated elements are not unified into special organs. They are dispersed elements that originally belong to the integuments with which they are in contact. Let's note that to completely clarify this question of undifferentiated elements and to understand their role in reproduction, it will be helpful to see if there is a difference between schizogonic properties and properties relative to the budding of the schizogonic germ. When it is notable in size, like the arm of a starfish or serpent star, this germ is incorporated into the new individual without rejuvenating it. This whole individual therefore has a part of its body that is old, while the other parts are new. During a new schizogony, does this old part have the same properties as those that have been newly formed? Can it still give birth by way of regeneration to a new individual? Systematic experiments in this sense do not seem to have been undertaken from the perspective of a study of neotenization. So here, maybe the key thing to clarify is this distinction between undifferentiation and dedifferentiation. So as far as I understand it, the distinction here is between so undifferentiated cells or portions of the organism are portions that have never been differentiated into specific organs or specific types of tissue. So they, from the early stages of development, are still in that undifferentiated state, whereas dedifferentiated tissues or cells would be tissues that were differentiated and then undergo a process of undoing that differentiation so that they become undifferentiated. So in some of these organisms, so the question is, when these buds arise in a hydra or other organisms that reproduce through budding, the question is which parts of the organism do those buds arise out of? So if we assume that there's some sort of strict distinction between soma and germen along the lines that Weissman is proposing, then there should be only specific parts of the body, like reproductive organs, that are capable of forming buds. Whereas if there isn't this strict distinction between soma and germen, then we should find that buds can form in various parts of the body of the organism. And what Simondon is arguing here in the case of the hydra, or following Rabot, who's arguing that even though there are specific tissues in which the buds form, those tissues are undifferentiated, so that they don't form something like a specific reproductive organ. It's just that there are certain local conditions that make it so that only those tissues can form buds, but there's nothing about those tissues that specifically assigns them a reproductive function, and therefore would make them part of the germen rather than soma. So yeah, we're still working on this distinction between soma and germen, and what the various modes of reproduction tell us about the validity of that distinction.
So I have kind of a general question about this breakdown of the distinction between soma and germen, which I think I understand this idea of hereditary substance from the last section. And the point he's making here about localization and how, like in the Hydra, as you were saying, it's not localized, which supports this argument that the individual is more substantial in the sense that it's more of a hereditary substance, which means like there's more of a unity of soma and germen. And I'm sure that he'll get into this later, but I guess I don't understand how useful this collapse of this distinction would be in describing more complex organisms, because it seems like there is, the more complex the organism, the stronger the distinction between soma and germen becomes. And there's only a relatively small part of the organism would be, or at least a localized part of the organism would be, hereditary substance.
Yeah, that's a good question. He has talked a little bit about certain certain organisms like some kinds of lizards, for example, that are capable of regeneration, and so like, for example, lizards whose tail can break off and then they can regrow the tail afterwards. And he distinguishes this from reproduction because the tail, of course, doesn't go on to regrow a full lizard. It's only the rest of the lizard that regrows a tail. And so in vertebrates in particular, you don't have reproduction through fission in the way that you do in certain invertebrates, like the hydras and so on that we've been talking about. So yeah, in that sense, there is a distinction between what we could call the hereditary substance and the rest of the individual, in the case of vertebrates and other organisms that can't reproduce through fission. But I think what Simondon is arguing here in relation to this distinction between soma and germen is that if it is valid, then it should apply to the living being as such, and independently of whether or not it's or whether those organisms can reproduce. So if there is really this independent, namely the germen, then that same immortal substance should be present in unicellular organisms as well as in multicellular organisms. Same substance, it should be sort of passed on through the whole history of life. Otherwise, we have to assume that there's some like moment in, I don't know, 3 billion years ago or whatever, when the first multicellular organisms arise, when suddenly this immortal germen appears, and then that same immortal substance has persisted until today, which is a kind of a weird hypothesis, and so I think it's that continuity of life, which is the basis for saying that if this distinction falls apart when you start looking at reproduction of single-celled organisms and certain invertebrates, then we can't apply it as this sort of strict substantial distinction in the case of vertebrates either.
Okay, thanks. That's really helpful. So it's more like he's not saying that this has to hold for, that this hereditary substance conception is true of vertebrates as well. His point is that this idea of the distinction between germen and soma can't be the basis for like the conception of vital individuals, because it's not true of some of these invertebrates.
Yeah, I think that's right. And I think we can sort of compare this argument to the argument that he gave in the part on physical individuation, where he he looks at the photon concept and the way that it is sort of useful and makes sense in certain frequencies when you're looking at the photoelectric effect with ultraviolet radiation. And so it's sort of the concept is useful in those restricted domains of the frequency spectrum. But then when you go to super long frequencies like radio waves, it starts to become less useful or less obvious how to apply this concept. And then he argues that if this photon concept is a valid, if it actually corresponds to something real, then we should be able to apply it across the whole electromagnetic spectrum. And so because it only has this limited application, it shows that there's the individuality of a photon is something only relative to frequency. So it doesn't sort of correspond directly to reality. And so he's making a similar argument here that if if the Soma and German distinction is mapping onto something real, then it should apply across the whole domain of the living world and not just to the restricted sphere of vertebrates. And so yeah, it's a I think he would characterize this as a transductive argument because you're arguing for or you're basing your argument on a certain homogeneity of a domain of thought or of discourse. So the same structures that apply in one portion of that domain should apply in other portions as well. And if they don't, that shows that there's some lack of reality to the concepts that are applied only in that one portion.
Thank you. That's very helpful.
So while reading this part, I thought of like the becoming like that is like from 10,000 Plato, chapter 10 something. So as far as I understand, like actually Toulouse must be influenced by Simondon as far as I understand, but the what I understood from the becoming, what is the exact title of becoming, but anyways like what I understood from the chapter and the discussion like the other day was becoming animal, becoming intense, becoming perceptible and so on. So that's kind of like overlapping some kind of a distinction like between should I say individuals, like some parts become overlapped and then that becomes like something new. Maybe we can talk more about like through those kind of becomings, like we can talk about a factor, whatever, things like that. But here Simondon is trying to like really scientifically explicate the process of kind of reproduction. That's kind of like one, for example, if there is one individual being, then that it has a kind of hereditary, some kind of distinctive features, and then that is kind of like the category of this differentiation. I may be totally wrong, so please correct me. And then those kind of differentiating features can be transited or transferred to other being, like by overlapping. I mean, some kind of features can be overlapped. I mean, here, like the word is used as a kind of bird or things like that. But like some kind of features can be moved to the other being, and then it can be some kind of like the point of reproduce some kind of like features in other being. And then undifferentiation is the moment like it can start to like totally, it is like a perfect kind of individual being, like it can reproduce its own kind of offspring. So what I want to say is like it seems like there are three phases, like differentiation, the differentiation, and undifferentiation. That is kind of a process of like something totally different to something similar, something same, like in terms of a category of a being. And then by doing that, it send some kind of features down to the offsprings, and then it did offsprings like a not just like a it could just as it's only one being, and then it can also how do I say, share some kind of features like with other beings, and then it extends like in terms of like the kinds of beings, like things like that. So question is that like undifferentiation, the differentiation is kind of can you say that is kind of three phases of like a process of extension of beings? I mean here like some kind of like the living beings, and the secondly there is word like localization, metabolism, something like that. It also gives me a question. I mean it makes me ask a question like metabolism, like in internal metabolism and external metabolism. Actually, it also like has to do with this process as well. So two questions like regarding undifferentiation, the differentiation. Like what I understand is like that seems like kind of a phases, like from one being to the other being. And then the second question is regarding localization or metabolism. So I think like it also function very, it plays a critical role in this kind of process. Sorry, like my question sounds really fudgy.
No, that's good. There's a lot in there. Yeah, so on the question of differentiation first, I think there's two different senses in which we can, well, at least two different senses in which we can understand the word differentiation. So on the one hand, it would mean the distinction between one organism and another. So each individual organism has certain characteristics, color or size or some chemical characteristics or whatever that are slightly different from every other organism, even of the same species. And so that would be differentiation in one sense, is just the difference between different organisms. But I think the sense in which Simondon is using it here is a different sense. So it's what we could call maybe developmental differentiation. So it has to do with the way that in the development of an organism, and this is specific to organisms that reproduce sexually, but you start with a fertilized egg, which is more or less undifferentiated. It's largely homogeneous in terms of its structure. Each part is similar to other parts. And then the early stages of the development of the organism, the egg separates and it divides multiple times. And it takes on the form of basically just a clump of cells that are, again, undifferentiated. So each cell is more or less the same as the others, and so there's no differentiation of the cells from each other. And so the differentiation, and then as the development proceeds, you eventually have differentiation within that mass of cells, so that certain, one end of the clump of cells turns into a head, and the other end turns into a tail, and so on, and the different parts eventually become different organs and tissues that have different structures, and so on. And it's actually, this is a very there's extremely complicated chemical interactions that govern these the differentiation of organisms, because each cell has the same genetic material, the DNA and the mitochondrial DNA and so on. It has the same genetic material, yet the same cells, cells that are basically the same and have the same genetic material will turn out to be, one will turn into a liver cell, one will turn into a neuron, one will turn into a muscle cell or whatever. They can turn into hundreds or thousands of different types of cells, and the way that somehow the cells know, in quotes, they know which what type of cell they're supposed to become in the developmental process, so yeah, that's like a whole field of research in biology, is just analyzing that process of differentiation through which an organism comes to be made up of different types of tissues and cells, starting from a clump of cells that are basically all the same. And so, yeah, so I think Simondon is referring to this kind of differentiation when he talks about these differentiation and undifferentiation and de-differentiation. And so when he talks about undifferentiation or undifferentiated tissues, so in the case of the hydra, for example, these are tissues that from the beginning of that process of development, they didn't undergo any sort of specialization and turn into I don't know what organs a hydra has, but like particular nutritive organs or circulatory cells or whatever. Like they just they retain that sort of undifferentiated form that they had at the early stages of development, and it's those cells are the ones that where the budding occurs, where that turn into the buds that split off eventually and form a new hydra. And then de-differentiation would be a process through which cells that are differentiated in some organ or some tissue sort of revert back to a form in which they don't have that differentiation. So they were differentiated, but then that differentiation becomes undone, and then they again take on that capacity to form all kinds of new structures, like to undergo further differentiation. And so I don't think they form like a sequence of phases exactly in that sense, like one after the other. So de-differentiation and undifferentiation are sort of alternate possibilities. So in some organisms, you'll have undifferentiated tissues that form the basis of reproduction through buds or something like that. And then in other organisms, you have some kind of de-differentiation process that occurs as a sort of preliminary stage of reproduction. So they don't happen in sequence in the same organism. They happen as alternatives in different organisms. Yeah, so I hope that answers the first question more or less. And then the second question, oh, sorry, go ahead.
Yeah, I like your explanation. That's exactly what I wanted to say. And then you really, really clearly said that. And then, yeah, it can be like said as something linear process. I mean, it's not a linear process of phases, something like that. But definitely I agree with that. And then thank you so much. That's a really clear explanation.
Yeah, and then the second question was about metabolism, and so the difference between some sort of internal metabolism and external metabolism. And so I'm not sure if Simondon makes, and then the question of localization, like what is the relationship between metabolism and localization? So I'm not sure if Simondon makes a a distinction between internal and external metabolism, but I think the way that we would sort of make sense of that distinction or try to apply that distinction would be like each cell within an organism has its own metabolism. It takes in nutrients from the environment in which inside the body. It takes in nutrients and it excretes waste, and so it undergoes a metabolic process in that sense, and that would be an internal metabolic process, and then the organism as a whole also takes in nutrients from its environment and excretes waste, and so it's that would be an external metabolic process. So something a cycle that goes outside of the body of the individual organism. How that links up with localization, I'm not 100% sure, but in the case of this hydra that Simondon is analyzing following Rabot, I think the localization issue is, so the question of or why we care about localization is that on Weissmann's theory that makes this strict distinction between Soma and German, we should find that that only one sort of localized or one particular portion of the organism has the capacity to perform reproductive functions. So we should find that there's a specific reproductive organ which in which the reproductive functions would be localized, and to some extent that is correct in the case of these hydras. So they form buds, but only from the specific tissues. And what Rabot argues is that even though it might look sort of at a first glance as if this is a case of localization, because these tissues are undifferentiated, we can't regard them as some sort of specialized organ of reproduction, and so we have to account for that localization in a different way. So we have to give an alternate explanation for why only these specific tissues form buds, and Rabot's explanation is that it has to do with the metabolism of the organism, like basically that certain chemical influences in the organism would mean that only certain tissues retain that undifferentiated state that allows for the formation of buds, whereas other tissues would undergo differentiation and wouldn't be able to form buds anymore. The localization is a product of the overall metabolism of the organism, in which certain chemical substances have different concentrations in different parts of the organism's body, and so that the overall metabolism of the organism accounts for this apparent localization of the reproductive functions, which Rabot doesn't want to analyze as being like a specialized reproductive organ, but as being functions of this undifferentiated tissue that it remains undifferentiated through the workings of the overall metabolism of the organism.
Yeah, thank you so much. Yeah, really helpful. And then metabolism, like actually you mentioned a little bit why that certain particular parts like react or like function in terms of metabolism. That's kind of the issue. And then my kind of like a random guess is that maybe that has to do later on, like a potential, the concept of potential. At the end of the day, potential of the particular part ignite. I mean, make it possible to localize. I mean, if I say things like that. So in that sense, like Yeah, your explanation, fantastic. Thank you so much.
Yeah, you're welcome. Yeah, so Angus has pointed out that we're getting up to the second hour now of our discussion, and we're still on the first page or so that we've read. So maybe we should move on to the next part, and that's not to discourage discussion, of course. So you know, keep the questions coming, but we'll try to move a little bit faster through the next couple pages. So if someone else would like to read the next page or so of the text.
I can read. We're at the paragraph after neotenization, I think, on 201.
Yes, I think that's right. So the localization of budding.
Okay. The localization of budding is also quite distinct in saltwater hydrozoans. In some hydrozoans, stolons form, which are non-differentiated buds. The cenosarc thins and ultimately separates from the original branch when the stolon spreads out. The parasarc thins in the bud, whether propagule or frustule, becomes free with a substrate to which it adheres and on which it slowly creeps. It is only at this moment that it proliferates out somewhat from its length. The proliferation rapidly grows perpendicularly to the longitudinal axis of the frustule, and 2 days later it transforms into a hydranth. The same frustule is in this way produces several hydranths that remain linked together. We should note that in this procedure of reproduction, there is a veritable synthesis of schizogony and budding. Indeed, the formation of the stolon begins as budding, but instead of proliferating, this bud detaches, which corresponds to a schizogony. Then the detached bud starts to proliferate, which corresponds to a budding. We should further note that this synthesis of schizogony and budding leads to a life form that is intermediate between pure individualization and a life that is so collective and with such strong bonds between individuals that the latter would no longer be anything but the different organs of a single whole constituting the veritable individual. Other cylinderates, such as campanularia, produce a frustule that, by detaching from the hydrocolus, drags along the hydranth under which it is formed. But this hydranth is reabsorbed and disappears as the frustule emits buds. Everything happens as if the activity of budding that generates a new ensemble were incompatible with the conservation of an already formed individual. Perhaps it is necessary to see in this disappearance of the hydranth a consequence of the D differentiation. We have seen it work in every reproductive activity, either via schizogony or via the formation of a bud. There is also budding in tunicates, where it is complicated by the fact that the bud develops at the end of a stolon and grows in the lower part of the body on an undifferentiated tissue that belongs to the mesenchyme, and is quite narrowly localized in the region of the post-abdomen. Maybe I'll read some of the next paragraph too. This stolon is a tube limited by the ectoderm and divided lengthwise into two parts by the mesenchymal partition. The stump of the stolon emits several buds, each of which grows and produces an independent ascidian. The active part of the bud is a block of mesenchymal cells originating from the partition. The entire individual is differentiated starting from these cells. The other elements are reabsorbed. In this case, the procedure thus conserves an aspect of budding. It is a budding at a distance that takes place through the intermediary of the stolon. Yet this is nevertheless a budding, since the separation only occurs after the differentiation. Budding presents itself in another form that poses the problem between the founding individual and the young individual. For example, in aquatic oligocate worms in the did I group. Indeed, budding occurs in quite a narrowly localized zone on the posterior part of the worm behind a septum. At this level, the elements of the external integument multiply starting with the ventral side. And what results is a thickening of this wall that propagates around the somite at the same time as a superficial constriction appears following the transversal median plane, revealing a relative morphological discontinuity between two individuals. The non-differentiated cells of the intestines multiply along with the elements of the mesoderm that line the segment's cavity. The various organs of a new individual, a zooid, differentiate within the embryonic tissue formed by these cells, with the head appearing in the anterior part of the bud in immediate contact with the tissues of the parent. Often this new zooid, before separating from its parent, in turn buds in the same way. It then constitutes a chain of several single-file individuals linked together one after another. Each individual buds in an approximately continuous way. It even happens that a second zone of proliferation occurs in one of the segments situated in front of the posterior segment. Moreover, it may happen that the undifferentiated zone is established in the last somite, higher up. Then the following somites, already differentiated before the establishment of the undifferentiated zone, do not de-differentiate in order to form the new individual. They immediately form an integral part of the zooid and unite with the homologous tissues originating from the bud. The founder regenerates the removed parts.
Yeah, thanks for the all the biological jargon. Yeah. There's one small point of translation, or well, maybe not that small, but it doesn't change the sort of big picture. But there's a missing negation in one sentence. So right near the end of that last paragraph, so where it says, moreover, it may happen that the undifferentiated zone is established in the last somite higher up. It should be is not established in the last somite, but rather higher up, something like that. So yeah, there's a missing negation there. It doesn't really change the big picture very much, but I just thought I'd point that out. Yeah, so I think so. We're looking at here alternate ways in which organisms can reproduce through budding or fission, and there's a sort of continuity from one to the other. So if we have like an organism that forms a small bud that eventually turns into another full organism and separates and goes its own way, we would call that a case of budding. But when we have an organism that separates and forms like two daughter organisms that are similar in size to the initial organism, we would call that a case of fission. But that distinction of like the size of the different products of the split is a kind of arbitrary one, and Simondon doesn't want to regard the size of the two components as sort of the criterion to use to classify reproductive forms. And so what he's looking at here is the relationship between budding and differentiation and de-differentiation. So in certain organisms, you have a bud that forms. So you have some sort of undifferentiated tissue, or whether it's sort of primitively undifferentiated, like in the case of the hydras, or whether it's the product of a process of de-differentiation. You have this tissue that forms a bud, and then the bud, while it's still attached to the parent organism, the bud undergoes differentiation and forms a new body that is differentiated like the parent body, and then the split happens. So you have the formation of the bud and then differentiation first, and then the split. whereas in other cases you have the bud forms and then splits off first, and then undergoes differentiation. And so that's closer to the case of schizogony in the sort of pure sense. Yeah, so the difference is the order between the split from the parent organism and the differentiation of the tissue into a new organism. So which one comes first? Yeah, so I think the sort of big picture is that these are more or less independent functions. That so they the differentiation into a new organism and the separation from the parent are independent to the extent that they can happen in either order. So one can happen first or the other, and so we have to, when we are analyzing reproduction, we can't take one of these functions as being sort of determinative and as being like the fundamental one, and the other one as a product of that one, so that we have to understand reproduction as involving both of these aspects, and to the extent that reproduction is characteristic of individuals as part of that individual function, as opposed to the colony function, then we would expect the split from the parents to be like the more fundamental aspect of reproduction, but we find that it's not actually more fundamental. They're both equally important or fundamental in reproduction in different organisms. So I think that's the sort of big picture of why we should care about the different worms forming of buds and so on. And so this reproduction through buds that differentiate first, like while they're still attached to the parent organism, and then undergo a split of some kind. This is characteristic of the colony form of an organism. So like these polyps or corals or whatever that form these colonies that share a circulatory system, they will separate. They reproduce new organisms that are still attached to the parent organism and that share a circulatory system. So they don't form a new individual in the strict sense, and then it's only in the cases where you have this strict or this sort of decisive separation of the parent organism and the child organism that you have something like a real individuation happen. Okay, so we can go on to the next bit if someone else would like to read from thus a definitively undifferentiated zone.
Thus, a definitively undifferentiated zone separates the individuals that remain aggregated in a chain. These individuals can remain linked together long enough to become almost adults. We find this in certain turbularians in the Rhabdocela subclass, which are non-segmented worms related to planarians. I cannot pronounce these words. We can see in this sense how the mode of reproduction is significant in the individual's relation to other individuals. The relation of dependence or independence expresses to a large extent the way the individual has been generated, which is why an important aspect of the inter-individual relation is a form of reproduction, even when it spans the whole life of each individual. This is what is particularly important to study in the case where various modes and degrees of individuation appear in colonies. Colonial budding is not always established following the same mode. In fact, there are all kinds of transitions between a proliferation that is nothing but a growth of substance and a proliferation that, after producing anatomically and physiologically distinct individuals, on the other hand, leaves them grouped in a mechanical unity. The two borderline cases can be represented in terms of ascidians and sponges. The ascidian represents the borderline case in which individuals, while separating from one another, nevertheless remain quite strictly grouped together. On the contrary, sponges represent the borderline case in which an active proliferation yields a simple growth of substance, whereas the new parts seem to be a certain number of individuals. However, even in this case, the individuated state is not completely nullified. It can appear if the mode of reproduction changes. The individuated state temporarily reappears if the sponge produces a bud that detaches. Something that occurs every now and again. This would confirm the hypothesis according to which there is a link between the appearance of the well-characterized living individual and the functions of amplifying reproduction. The individual is essentially the bearer of the capacity to reproduce, not necessarily to reproduce itself, for it can, on the contrary, reproduce a colony that is not at all comparable to it. As we have seen, the reproduction of ascidians is performed by a stolon. The stolon expands while anchoring onto a substrate, and then its extremity develops into an individual that detaches from its founder but remains anchored in place. All the stolons originating from the same founder behave in the same way and produce a certain number of buds. What ensues is a grouping of individuals anchored side by side but independent from one another. Conversely, the sponge, which is at first simple, begins to ramify, and each ramification takes on the aspect of the initial sponge, with a new osculum and inhalant pores. Morphologically, these new parts seem to represent a series of individuals, but the external morphological criterion here is lacking and is shown to be insufficient. These ramifications remain in complete and definite continuity with the sponge's mass. None of them have the value of a bud. The various regions of the sponge form a new mass, each comprising part of the whole in which no element possesses a veritable autonomy. Let's note, however, that the ensemble of the sponge can hardly be called individual more so than each of its parts. The various parts are not organs of the individual that would be the sponge, since these various parts are not merely continuous but also homogeneous. The appearance of new parts is a growth of the quantity of the sponge's living matter, but does not bring about a significant differentiation. Since there is not more in the whole than in the parts, it is difficult to call the whole an individual simply because it is the whole. The whole is not indivisible at all. Not indivisible at all. If we remove a part of the sponge that multiplies, the sponge is not mutilated but diminished. Here we are faced with an absence of structure that does not allow us to give the name individual to the whole more so than the parts, nor does it allow us to remove this name from the parts in order to give it to the whole, since the whole is nothing but the sum of its parts, the heap they form. In fact, this extreme case is the one in which individuality belongs equally to the parts and to the whole. The parts do not have a veritable individuality since they are not independent, but they nevertheless have a defined form with an osculum, inhalant pores, and a certain orientation with respect to the whole, an orientation that is more prominent in certain species. Thus, there is no absolutely complete continuity between the various parts, and a relative unity belongs to each part despite their lack of independence. Each part is complete by itself and could be self-sufficient. Each part, therefore, possesses a certain virtual individuality that the mode of reproduction does not highlight. Furthermore, the whole also possesses a relative individuality that is complementary to that of the parts. This individuality consists of the rudiments of orientation that seem to direct the genesis of new parts. They do not come absolutely at random from the old parts, but according to certain directions of privileged growths. The studies that have been carried out until now are not sufficient for us to say with certainty by what force the whole acts on the parts so as to orient them, which thereby produces, despite the randomness of proliferation, ensembles that are not organized but ordered. The first degree of individuation prior to which there is nothing but pure continuity. Indeed, what is quite remarkable is the fact that the individuality of the whole only appears here as a form and not as an organization. But this existence of a form is not negligible, since the individuality of the whole consists precisely of what is removed from the parts in terms of their freedom and their capacity to grow in all senses. However slight this influence may be, it is nevertheless a subordination of the generation of the parts and of their growth to the existence and arrangement of the whole. It is the origination of a structure. The slightest appearance of individuation is therefore contemporaneous with the manifestation of a dynamic structure in the process of a being's reproduction. A reproduction which, moreover, is still not distinguished from growth.
Yeah, and again, he's going into some of the details of the specifics of reproduction of these different organisms that we probably don't need to worry too much about. But he's representing these two cases, the clavulin, whatever that is, and the sponge, as these two extreme cases. And I think the translation here is not quite correct. It calls them borderline cases, I think, if I remember correctly. But anyway, yeah, so it uses the word borderline case, but I think what is actually meant here is an extreme case. So the sponge and the clavulin are two extreme cases of reproduction through budding and what that can look like. So in the case of the clavulin, we have the formation of a bud which separates from the which first attaches itself to the substrate, to the bottom of the ocean floor, I suppose, and it becomes fixed in place, and then it separates from the parent organism, and so you get like a row of these organisms side by side, but they're independent of each other, so they form sort of individuals in that sense. Whereas in the case of sponges, you have budding without separation, and actually, it's not even really budding in the proper sense, because it's just growth, and then the formation of a new set of pores, and so it doesn't take on the form of a bud that has the capacity to undergo further differentiation. It's just growth in space, and then the formation of these new pores and so on. And so in the case of the sponges, there's no sense in which there's any sort of individual in question. So the parts of the sponge are not individuals, because they're incorporated into the whole sponge colony. The whole sponge colony is not an individual, because it's just made up of all the parts. It has no structure really of its own, and the only sort of qualification of that, the only sort of beginning of a structure that appears is that the shape of the colony is not random, even though each part of the colony sort of reproduces at random just by growth, and the overall shape of the colony is not random, so that it retains a certain overall form, and so the state of biology at the time that Simondon was writing doesn't really allow to explain what is behind this overall structure of the colony, and I'm not sure if this has been explained since then, but yeah, there's just this very general or very superficial structuring principle, which means that the overall growth of the colony takes place in some sort of overall form or shape. But yeah, so these are two sort of extreme cases of the forms of growth in relation, or the forms of reproduction in relation to individuation.
So that's a 203. What you are talking about is like the paragraph starts, which starts like colonial budding, such and such, and then the two borderline cases, not the two borderline cases, but the two extreme cases, which means like in between there are more stages or states, right?
Yeah, so here he only talks about these two extreme cases, but yeah, we given that he says there are all kinds of transitions between a proliferation that is nothing but a growth of substance, so that's the case of the sponge, and a proliferation that, after producing anatomically and physiologically distinct individuals, on the other hand, leaves them grouped in a mechanical unity, and that's the case of the flavolins, which I have no idea what exactly that is. Yeah, so there are other cases that are in between these two extreme cases, so some sort of intermediate form. Yeah, thanks. So Angus has posted in the chat the Wikipedia page for Ascidians, or the Acidea order, or whatever class. Sorry, and yeah, so there are these weird underwater organisms that have these tubes that they filter stuff out of the water, and feed on whatever they can filter out of the water. Yeah, so that's what an Ascidian does. Yeah, so let's go on to the next bit here. Where did we finish? Right, so we're at the top of 205. I'm not sure if we'll have time to finish this chapter today. We might just have enough time. But yeah, I'll read the next page or so. Furthermore, let's note that if two sponges are very close to one another, the budding that they emit indeed lacks a distinction between the two groups' individuals. This dynamic structure of growth does not pass from one individual to the other. The extensions of each sponge remain distinct and do not influence one another, as if this morphological dominance exerted by the whole onto the parts were reserved for them alone and would not be transmitted even by the narrowest proximity. The morphological criterion is therefore important, since it appears at the very first degree of individuality, in a state in which individuality is still fragmentary and does not exist in the whole, except in the slightest perceivable way. Everything happens as if individuality were a measurable physical parameter, or quantity with the ability to distribute between the parts and the whole. The more the whole is individualized, the less individualized the parts are. On the contrary, if the parts are almost complete individuals, virtually detachable without requiring regeneration afterwards, the whole is poorly individualized. The whole nevertheless exists as an inhibitor or accelerator of the growth of the parts. It plays a morphological role through its dominance, which is exerted on reproduction. We must regret that studies on the genesis of forms have not been pushed far enough for us to say by which agent these accelerating or inhibiting influences exert themselves and constitute a veritable field of growth in which the individual develops and which it itself involves. The same type of phenomena is present in the vegetal world. Lichens, an association of algae and fungi, do not develop anarchically. In certain species, the extremities are calloused and endowed with a certain hardness. When light is not very abundant, forms become comparable to those of plant leaves, such that this vegetal association could be mistaken for a single living plant in the same type of milieu, for example, sedges and ferns. Between the two extreme forms of Ascidian and the sponge, there are a multitude of degrees of individualization of the ensemble, i.e., according to our hypothesis, a multitude of values of rapport between the degrees of individualization of the parts and the degree of individualization of the whole. Other Ascidians produce stolons arrayed more or less regularly, but without their own membrane. These stolons ramify and tangle together in the particularly thick membrane of the parent, and then bud within this membrane. The buds partially emerge while developing. The region of the thorax, which includes the pharynx and the peribranchial chamber, has its own membrane and emerges from the parent's membrane. Once they have fully developed, the adults remain in continuity with the original stolon but lose all functional relation with it. Only the shared membrane joins and maintains them. There is, however, a certain regularity to the grouping. The mere fact of having a membrane and more so a common origin suffices to define for all these developed buds an incorporation in the individuality of the whole. Since each individual buds in turn, the colony, which envelops the products of several generations, branches out and can acquire fairly large dimensions. We should nevertheless note that this dynamic structure of the ensemble seems to have a certain limit. The whole colony is not organized all of a piece. When it is large, it is formed from several groups distributed at random, but each group represents a certain order. These groups, which truly indicate the dimensions of the group's individuality for the species considered, are called coenobia. So here is just the point that we were just discussing a little while ago, that there are intermediate forms that are between the case of the ascidian and the sponge, which are the extreme cases. So we have certain other forms, other ascidians that reproduce new individuals, but or quasi-individuals, but they don't have their own membrane, so they reproduce inside the membrane of the parent individual, and they produce some sort of tangle of these different individuals within the membrane. And then, yeah, so they have a shared membrane with the parent, but then they have no functional relationship with the parent anymore. So they don't have a shared circulatory system or anything like that. Yeah. So then we have, and he also points out that similarly to the case of the sponges, we also have in certain plants and in the case of lichens, and lichens are extremely weird sort of beings. You can't even call them organisms because they're an association of two different organisms. So it's a algae and fungi that sort of grow together, and that allows them to the association of these two organisms allows them to live in environments in which neither of the two associated organisms could live on their own, and they've co-evolved through this association so that they can live on like bare rock, for example, where you couldn't find the alga or the fungi on its own. Yeah, so that's sort of an aside, but yeah, you have a similar form of growth. So the lichen just grows sort of in size, and it doesn't split off in the way that it doesn't form buds and split in the way that other organisms do. But it doesn't. It still has the total group of lichen has a certain form, and so in environments with low light levels, it forms something like a leaf that looks like a plant leaf, and which helps to absorb more sunlight. So the total group of lichens, or the clump of lichen matter or whatever, is not just a sort of random association. It has a structure at the level of the whole. So it's a sort of beginning of individuation at that level.
Day, like nothing comes up all of a sudden, right? Like out of the blue. Everything has a kind of a continuity. I mean, in terms of species, everything like has a continuity, right? Like it comes after and after, it passes down and things like that.
Yeah, so that would be true, or that would be largely true in evolutionary history. So that each each organism inherits from its parent organism most of its features or properties or whatever you want to call them, so that most organisms would be similar to their parents in most respects. But what can happen is that in the evolutionary history, you can have say 2 extreme forms, and then all the intermediate forms can die out, so that you only have the extreme forms left over. So like it could be at some point in the future, we might only have these ascidians and sponges and not have any of the intermediate forms. And then there would be this sort of a gap or a jump in terms of the degree of individuation from the sponges to the ascidians. So that possibility of certain organisms dying out means that you can still have something like a gap or a jump in terms of the properties of organisms. Yeah, and the whole issue of intermediate forms, this was like when the theory of evolution and the Darwinian theory in particular was being introduced and debated. This was one of the points that Darwin himself recognized as being a sort of a weak point of his theory or a difficulty for his theory, was that it was the absence of intermediate forms between certain types of organism, I'm trying to think of a good example, but well, in the case of humans in particular, this was one of the difficulties at the time. So before most of the fossils of early hominids that we now have were discovered. So Darwin, of course, argued that humans had a shared common ancestor with apes, but there's no like sort of half ape, half human organism or something intermediate between an ape and a human that was known at that time, and so this was a sort of difficulty for the theory, and it turns out that of course there were other hominids before modern humans that in some respects are closer to today's apes in terms of brain size, for example, but they also resemble humans in other respects, like the bipedal posture. So yeah, there was a sort of a search for the issue of intermediate forms. So the fact that the intermediate forms had all died out means that there was a sort of gap in the continuity of nature.
On page 204, like from the bottom, 1, 2, 3, 4, around like 10 lines from the bottom, there's what force the whole acts on the parts so as to orient them, which thereby produces, despite the randomness of proliferation, ensembles that are not organized, ordered, blah blah, and the continuity. So actually, this kind of what force like makes everything possible. Still, we don't know, right? We don't know about that, right? Just like what I think from this part is like Simon seems to just like advocate the idea of like there's no missing links, like in terms of continuity, like in terms of like evolutionary theory, right? But still, like we don't know exactly what force make everything possible.
So when he refers to continuity here, he's referring to organisms like the sponge, where actually, well, sorry, it would be something even less structured than the sponge. So the sponge has the first sort of stage or the first sort of glimmer of individuation, in that the whole colony of sponges has a certain overall shape, and so we could we could picture, I'm not sure if there are any organisms like this, but an organism that would be like a sponge, but without having anything like a general shape of the colony, so that the aggregate of the organism would just grow sort of at random, and there would be no sort of overall shape of the aggregate. That's what he means by continuity. So it's organisms that don't reproduce new individuals; they just grow in size. And then he says that the sponge, the way that the sponge colony has its determined shape is just is the first degree of individuation above the level of just pure continuity, so that it's that structure of the whole, or that shape of the whole, that is the first degree of individuation above the degree in which there would be no individuation at all. So in this case, we do have the intermediate forms, like they do exist, but that's a contingent fact. It's just it happens to be the case that the intermediate forms didn't die out historically, but it could have happened that the intermediate forms died out, and we would have like a jump in the degree of individuation from zero individuation to a much more strongly individuated organism. Okay, so I think we might just have time to finish the whole chapter if we sort of read through everything in one go. If someone else would like to read, I think we're at a similar process of reproduction on page 206.
Let me read.
Sure, go ahead.
A similar process of reproduction takes place in heterocarpa glomerata, which generate stolons that are reabsorbed when the new individual has been born. A single membrane remains that strictly maintains the products of several successive generations tied together. Here again, the mode of reproduction is indeed what determines the specific degree of individuality, linking the regime of individuation back to that of reproduction. Reproduction in golden star tunicates, Vortellus, takes place in a different way that leads to a different regime of individuation. Reproduction takes place through a very short stolon, whereas in Polystyrlenes, it grows to 1.5 centimeter that integrally transforms into an individual. The buds then form clearly delimited synovia. The whole colony derives from the first individual that begins to bud before having reached adulthood. Afterwards, this budding occurs systematically until 4 birds of the same generation alone remain, those that would support them having been reabsorbed. These birds are arranged into crosses in such a way that their closely converge the colonies into a shared. How do I read this one? Cloaca, right?
Cloaca, yeah.
Cloaca, around which, thank you. The successive generations are grouped as, and when older generations disappear, where reject is an important agglomeration of individuals that completely possesses all the organs, most notably the heart, that make an autonomous life possible. However, the autonomy of the individuals is not full-fledged. They conserve vascular relations between each other. A circular vesicle, surrounds the synovium. Each individual has a heart whose beating is not synchronous with the beating of the others. In this sense, there's a regime of reproduction in which a clear morphological dominance of the whole over the parts manifests by way of a fairly rigorous symmetry in budding, and then by way of a circular form of the synovium during its development, corresponds to a colony in which the individuality of the whole is quite clearly prominent to the point of creating vascular relations between the individuals. Continue?
Yeah, you can continue. Yeah.
In Slant-rates, the formation of the colony is a frequent phenomenon. The majority of hydroids produce a large number of stolons that arise beneath of the hydrants and then stretch out and ramify without detaching from the founder. By ramifying, they emit lateral buds that transform into hydrants and in turn sprouts a stolon. This ramification is indefinite, and an equally indefinite colony corresponds to the indefinite process of reproduction. We should, however, note a very important but poorly stated fact in order to be able to found a theory on it alone. Certain ruptures are produced in this indefinite ramification that lead to collective individuals and limited colonies, as in the previous cases, in which we saw the colony produce via proliferation that a single colony of indefinite dimensions, but synovia of limited dimensions. Everything happens as if a certain quantitative limit produces an elementary morphological induction that divides the colony into restricted groups. A certain phenomenon of individuation, therefore, seems to arise even within the process of growth, which here are not separate from those of reproduction. Hubble considers these ruptures as accidental and not physiological. He separates them from the ruptures of short stolons, which he qualifies as physiological ruptures. But the conditions of these physiological ruptures are as poorly understood as the ones that interrupt the continuity of development. Thus, there is no irrefutable reason that forces us to oppose so-called accidental ruptures to physiological ruptures. Perhaps they depend on one another in the same way as the processes of reproduction considered, and its a dynamic structure which presides over the establishment of the anatomical or physiological structure of the colony or groupings of individuals. Within one of the groups of hydranth, Sinosauria remains continuous along the hydrocolus, thereby putting into relation all the hydrants through the system of channels that crosses it. In this sense, certain physiological bonds and the nutritive community in particular are established by this morphological continuity. There is itself accompanied by a continuity in the process of reproduction. And yet, indirect nature of this continuity leaves the hydrants with a certain degree of functional autonomy. Continue?
Yeah, we just got a couple paragraphs.
Sorry, this is alarm. The form of the colony is in general correlative of the mode of reproduction. Thus, in other colon traits, like hydrocynes, the stolon clips along the ramifies while remaining strictly in contact with the substrate. This is how it forms a network without any erect branches. The buds are born and grow perpendicular to this network, thereby transforming into elongated hydrants. How do I do? Continue?
Yeah, we just got one more paragraph left in this chapter, so let's continue.
In Hexacoralia, birds arise directly at the expense of the wall of the body of the skeleton that serves as a point of support. Colonies take on quite varied forms, but these forms are in relation with the mode of generation, thereby allowing for the recognition of species. In the immense colonies of madropores, which form coral leaf, the existence of polarity is quite prominent. Development often takes on the form of extremely ramified branches that follow the orientation of the whole, which indicates a relative morphological individuality of the colony. The aesthetic aspect of these coral ramifications seems to indicate that this morphology is not arbitrary. This morphology could be likened to the way in which certain complex efflorescences form, like those of ice, which is not independent from the characteristics of the substrate on which it forms, which it deploys forms in harmony with the laws of crystallization. Perhaps it would be necessary to seek in the kinship of form the functional analogies that link together a large number of processes of individuation belonging to extremely different domains. One aspect would be shared by all, the identity of the process of growth, which would be the creation organized ensemble based on the self-constitutive schema that responds to a dynamism of growth and to the initial givens depending on chance. The same law could then be found again in the growth of the efflorescence, in the development of the tree, in the formation of a colony, and even the genesis of mental images, as if the dynamic dominance would give structure to ensembles based on the singularity. A morphological analogy could reveal the identity of the process of formation of collective individualities. In each case, the structure of the individual would be linked to the schema of its genesis, and the criterion, perhaps the very foundation of the individuated being, would reside in the autonomy of this genetic schema.
Yeah, thanks. That was a long bit with lots of biological terminology that I don't really know exactly what it means. Talking about these different species of organisms and their structures and so on. So here we're still going through some of the different modes of reproduction of these different organisms, and the relationship between the modes of reproduction and the degree of individuality of those organisms. You have these golden star tunicates that that reproduce through forming the stolon that transforms into an individual, so you have a whole colony that forms from out of buds of starting from one sort of founder individual, so the colony takes on the form of a cross, because in each generation there are four individuals or four buds that form and form these new stolons, and so the colony has a cross form, and and then the older generations die while the elder generations are still alive, and so there's a overall form of the colony, but each individual has its own function as a living being, including its own heart. But they also have some functional relation to each other. So they have a shared circulatory system, but each of them has its own independent heart. So they nutrients and waste and everything circulate throughout the colony, but each individual within that colony has its own heart that pumps on its own rhythm, and so they aren't synchronized with each other. So this is an intermediate stage. So you have what are to some extent individuals, because they have their own heart and their own vital functions, but they also are incorporated into a larger grouping that has its own structure, that cross shape and the shared circulatory system. And then you have other forms, other organisms that have this sort of indefinite formation of a colony. So the colony doesn't have this rigid structure like in the case that we just saw with the cross shape. So the individuals in these hydroids, they form buds more or less at random or sort of indefinitely. They don't have a specific structure, but it's not actually strictly random. Like if you look at the shape of a coral reef, there's a sort of branching structure, which again is not a fully random. They don't just appear randomly, but there's a sort of spreading out of the organisms, and it's you can compare it to the branches of a tree. That the split of a branch is not random. It's the branch splits in such a way that the surface area is maximized, so that as much surface area is available to absorb sunlight as possible, and so you have similar sort of branching structures in the case of these corals and so on. And then he even I'm just going to skip ahead to the last bit. He even suggests that the association of ideas, or the genesis of mental images, has a similar sort of branching structure as the growth of a coral colony or the branches of a tree, so that there's some sort of shared schema or overall structuring process that we can find in the growth of a coral colony, or in the development of mental images from one image to the next, and that sort of branching structure. And so it's that schema of genesis, which is sort of characteristic of an individuated being. So it's the fact that there's something like an overall schema that is what characterizes an individuated being, as opposed to just a collection of entities. But yeah, so he gives us this sort of hint towards the psychic individuation that we'll get to in the next chapter, or the next part of the book, I should say. And the relation between vital individuation and psychic individuation.
I guess maybe that in next chapter we can also figure out what singularity at the end of the part of today, like singularity or the autonomy of this genetic schema, something like that, regarding kind of a force. Even though you explained it really well, still I can't figure out what force makes everything possible. So I think that has to do with the singularity or the autonomy of the schema, the genetic schema, something like that. Maybe you can figure out later, in relation to, as you said, the psychic individuation process, something like that.
Yeah, I think that we'll have a better sense of what this schema means when we get to psychic individuation. So yeah, it's a good question, but let's hold on to it and try to answer it when we get to psychic individuation. Yeah, so we're at time, so let's end here for today, and we'll pick up from page 208, so the beginning of chapter 3 of this part next time. So yeah, thanks everyone. Yeah, I think we had a really good discussion today, and some really good questions. So thanks, and see you all next week.